Everyone. Good morning, everyone. Welcome to the sixth ASAP webinar on modeling and simulation. Today, it's my pleasure to have both bSpark Bio and Novasign speakers here as guests. Yeah, and it's which is quite nice because both companies are quite experienced in conducting collaborative projects within ASAP. Yeah, and the two speakers today will present a case study, and this was actually partially conducted within ASAP project. Yeah? But first, I wanted to give a short outline on how the webinar is structured. So each presenter will have a 20-minute presentation approximately. Then we have about 10 minutes for Q&A. As you heard, we are recording the meeting. So the Q&A session can also be done anonymously. At the bottom, you see the Q&A button where you can ask questions also in anonymous mode. But you can also write it with your name revealed, and you can also raise your hand, and then I can get you into the panel and you can ask the question online, basically. So after 1 hour, we will stop the recording, but if there's still interest in discussion, of course, you're welcome to stay and the speakers have time. Yeah. So, but without further ado, it's my pleasure to introduce Didi Palmberger. He's a CTO and co-founder at bSpark Bio. And with this, I hand over to you. Thank you for coming.
Yeah, thank you very much, Nico, for your kind introduction. I'll try to share my screen and hope that everything works as it worked a few seconds ago. Can you all see my screen?
Looks great.
Work? Good, perfect. Yeah, before I'll start going into details about the process development of viral vector-based medicines, I would like to give you a short introduction about bSpark Bio. Who are we and what are we actually doing? So bSpark Bio was founded around 2.5 years ago by myself and the two co-founders, as Nico already mentioned. And we are heavily dedicated to the development of manufacturing processes for novel biotherapeutics. And during our past professional career of, depending on whom, 10 to 15 years, we already worked together on several development projects and brought products, mainly viral vector-based products, into the clinics and up to phase 3 readiness. And during that project, we, let's say, also took decisions that were suboptimal and that led to redevelopments, etc. So I think during that time, we gained a lot of knowledge on what is really necessary to bring a product to the clinics or to have a streamlined process development. And by that knowledge, we somehow came up with 4 pillars where we think those are important for efficient development project and for being able to survive that what we call early valley of death in the process development. And the one or the very first pillar is the strategy development itself, meaning before you start the whole CMC project for a specific product, it's really important to do a thorough roadmaps, development plans, to think about a possible risk management, to think on a regulatory strategy and on quality considerations. So this whole CMC strategy development is not only done by the process development experts, it's mainly a joint effort of the product developers, of the clinical team, and of the process development team. The second big part is the process design and the process development itself. And the first point here is the phase-appropriate process development. So it's not necessary right from the beginning to develop a manufacturing process that is ready for commercialization. But the process you design, for example, for clinical start, should have a solid foundation you can use to build on for the next stages. It's also very beneficial to have a certain degree of process understanding. And the process you develop should also be capable of supporting the doses you need for phase 1, phase 2, for phase 3. And a very complex thematical complex is the formulation development, about storage, about buffers. How can I store my products? The third pillar is the process analytics. So after you have produced your vaccine or your gene therapy product, you need to release it for market usage, for example. And for every product you have in your European pharmacopoeia, you have specific requirements in there, quality-wise. And so your analytical toolbox really needs to be capable of proving that your product fulfills the required specifications, either by the pharmacopoeia and also by the specifications you put on your product. And the last pillar is the GMP manufacturing or the GMP support. So, and that's really something you have to be very wise and clear on selecting your contract manufacturer. You have to transfer your process there, you have to do manufacturing oversight, etc. And it's really important to, let's say, screen a lot of CDMOs, and not only from a technical perspective, but also from a personal perspective. Because it's almost like in a marriage. If you sign your contract with a manufacturer, you are locked in. And if you then find out that things are not working as planned, that technically it's not working, that you're not able to work together on a personal basis, it's very hard to get out of that contract anymore and switch to another organization. So really choosing wisely is critical here. But today we'll mainly focus on the process design and the process development itself. And before we start here, I have a general question to the audience. Who of you is, when you start a development project for a product, who is thinking of the first steps, how to plan them, how to do them? And who is more thinking of the first step and already the second step and the third step and the fourth step right from the beginning? Just maybe raise your hands.
But did you raise your hand for what?
Just raise your hand if you think, if you already think some more steps ahead, or if you mainly think for the first steps when you start your development project.
So we have only 2 hands up so far.
Okay, one is your well,
now it's only 2 counting mine as well, but before it was 2 in the audience. So I don't know. It's just a show of hands. I'm not going to call anyone out. So if anyone else,
okay. Yeah, that is actually something we already realized when we're doing projects with our clients, and I like to compare the start or the process development itself with crossing a pond or a lake or a river using stepping stones. Because you usually start at the beginning and then you have a look at your next step. You take some decisions. Then you're going to your next step, take some decisions, and this is going. But in some situations, then you find yourself in some kind of a dead end. So you put a lot of efforts into your development, but then you're somehow stuck. And that can lead to the fact that you have to go back to the beginning, redevelop your whole manufacturing process, which is enormously time-consuming, heavily costly, and can, in the worst-case scenario, lead to the fact that the whole campaign is stopped or the whole product will never make it to the market. So it's really important right from the beginning on to, let's say, take the right decisions, or at least to take the decisions in a way that you can build on them for being able to have an efficient, robust, and scalable manufacturing process, and also for a cost-effective manufacturing process. It's not necessary to plan every step thoroughly and perfectly up to the end. But at least, let's say, the most critical decisions have to be taken right from the beginning. And then you have a solid foundation you can build on for phase 2, phase 3, and even for commercialization without the need to redevelop everything. When we now have a look at the viral vector-based medicines in general, and I picked two distinct areas and their challenges. The gene therapy, for example. I mean, the gene therapy in general at the moment is a bit of a let's say, complicated and tricky task because many companies are closing their efforts on the gene therapy. In general, it's still limited to rare and very ultra-rare indications. And one reason among them is for sure that the manufacturing costs are incredibly high. We have manufacturing costs per dose of several $100,000. And so for getting them, or let's say for supporting to get them out of the rare and ultra-rare indications and make them more broadly applicable for other applications, the efficiency in process development actually needs to increase by almost up to 100-fold. Because by increasing that, we can get the manufacturing costs down, and then at least the costs of a gene therapy treatment are cheaper. Because at the moment we are at costs per dose, or costs what a dose is sold of $3 million up to $3 million, which is insane. On the other hand, we have the viral vaccines. The viral vaccines are the total opposite than rare and ultra-rare indications, because when we think of the COVID pandemic, we need billions of doses in the shortest period of time. And for being able to also supply the LMIC, so the low and middle income countries, the manufacturing costs can only be in the lowest single-digit US dollar range per dose. So the challenges in process development here are that we need to be highly efficient, we need to be fast, we need to be scalable, and the process also needs to be robust because we want almost every batch to be successful in order to be able to feed the market. And what we learned, I mean, we have been working on client projects in the field of gene therapy and also in the field of viral vaccines, of oncolytic vaccines, etc. And what is surprisingly common among all of them is that the development, when we started to be involved in the projects, are mainly based on some kind of a shotgun approach. So they had one parameter, the parameter was varied, and they monitored the outcome. And then there was a next parameter that was varied, and they monitored the outcome. The big advantage on that strategy is actually that there is no prior knowledge required. So you can really start it from base. But the problem is it's rather unstructured, and it fails to really generate in-depth process understanding. So it's quite useful to, let's say, set on the concept of quality by design quite early on in process development. So the QBD principle itself ensures that the desired quality of a product is given by assessing variables which might impact the quality of a product. And this is done by a systematic approach and by combining the science on one hand and risk management on the other hand. There are several ICH guidelines on that, and it's not necessary to implement everything right from the beginning, but at least the basic concept should be established at the beginning. And the basic concept starts with the definition of a product profile, a target product profile or a quality target product profile. To get there, this usually combines the clinical team, the product development team, and the CMC team, where you define what's the real mode of action, for example. What's the route of action? Should it be intravenous injection? Should it be a pill? The next step is to think about critical quality attributes, meaning which parameters of the product itself are critical. For a gene therapy product, this can, for example, be the full to empty ratio. Or for viral vaccines, this is the virus titer, the host cell DNA levels or the host cell protein levels. Then you break it down even more. You go to the critical process parameters, meaning which parameters of your process critically impact, for example, your virus titer or your levels of host cell DNA. Based on that, you can then define a design space where your process should operate in and where you have a certain knowledge and where you can guarantee if your process is operated in that space, it will give you a product that is always within your specifications. After you have your design space, you can define a control strategy and you can do your continuous improvements. So the question is how to get to all those things. Well, you need to have a basic process established by whatever way. And then you actually lock yourself for several days in seminar rooms in London, for example. You break down your process in every single step and rank it regarding likelihood that this happens, severity if it happens, and then you come up with a, let's say, science-based risk analysis and a criticality map, where you then really get this parameter is highly critical because it might impact that critical quality attribute. So from going from this classical one factor at a time approach to a QBD approach, you can really get a heavily increased process understanding on the one hand. And on the other hand, you reduce your risk of failure because you really understand your process and you know what's going on. The big problem with that approach is that you need a baseline process already established. And for being able to get there, this is quite work intensive. And that's the reason why we, let's say, come up with a strategy, what we call for ourselves is advanced development, where we are combining pre-established building blocks, and a building block can be a unit operation or can be a set of unit operations, where we have a quite good prior knowledge on critical process parameters, and we are then combining that with machine learning data models. And I would now like to present you a case study that we did together with Novasign. And I will give you a little bit of an overview about the process development perspective. And then Max will give you an overview about the design of DOE and the modeling perspective on that case study. So the process itself was for a viral vector-based medicines. It was in viral cells. Cells were grown, bioreactors were inoculated, and the bioreactors were then infected with a virus stock. And then there was a clarification, purification, and concentration. Today we'll mainly focus on the upstream process. So we, many years ago, and this project also started at the cooperation with ACIP, started with a classical evaluation by a one factor at a time approach as a company that is not thinking about modeling or everything. As a classical startup is starting a process development. And the first parameter was we have a bioreactor, we're working with viral cells, and we want to know which microcarrier density might be favorable for producing the virus in that culture. So we varied the microcarrier density even to higher densities and had a look on the cell densities we're getting there. And we clearly saw more microcarrier meant more cells in the culture. The next step was due to the fact that those are adherent cells growing in microcarrier. This whole sampling procedure is a bit time consuming. We thought of why not implement a PAT sensor for being able to give us a bit more real-time information on what's going on in the culture. That's the reason why we implemented a viable cell density monitoring using a capacitance measurement. And that actually enabled us to get already a bit more in-depth information about what's really going on in real time. We can have 1 grams per up to 3 grams here, and we have a nice correlation of cell growth, of the capacitance signal to cell growth in the bioreactor and the cell density in the bioreactor. And the plan at that time was to use that signal as a trigger for infection timing and for optimal harvest timing. Then we went further and we wanted to see what's the kinetics of virus production. We clearly saw that the longer the time, up to 6 days, the virus titer is increasing. But we already saw is when we do that at different microcarrier densities, that it seems that the microcarrier density is somehow also impacting the virus titer we're getting out from it. But not in the way we thought. Because at the very beginning, we thought more microcarrier in the system, meaning more cells in the system, meaning more virus. But in the end, this was not true. We had way more runs, those parameters, I just have in the graphic here, those runs that were really run at exactly the same conditions. And what we surprisingly found is that at a certain microcarrier density between 1.5 and 2 grams per liter, we got a plateau with the highest virus titers, versus at 1 grams and 3 grams, we're almost at the same level. Meaning if you are, you can run your process at the 3 grams, but this, for example, has two negative effects. The one effect is that your titer might be lower, and the other effect is inoculating a bioreactor with a higher surface, you need, at the same density per square centimeter, you need a way higher cell density, which means this puts more pressure on your seed train. So if you are reducing the density, your seed train gets easier, and you also have the benefit of increasing your virus titers. So really thinking not only on one unit operation, thinking out which impact that might have on the unit operation before and the unit operation after that. Because more cells also means more host cell DNA, more host cell impurities that the downstream needs to remove in the end. So balancing between virus titer, seed train complications, and downstream. Those are all critical thoughts you have to take. Yeah, and then we had some kind of this baseline process. We did a control strategy. We identified 3 potential CPPs. We defined a design space with around 10 bioreactor runs, and we did a DOE with 7 bioreactor runs of 4 bioreactors each. So you already see that this is quite a lot of work for getting into a solid understanding. And we actually got out with a first result, and the 3 CPPs we thought that might be important were temperature, MOI, and pH. And we clearly saw that temperature, yes, temperature has a clear impact. The higher the temperature, the lower the virus titer, with an optimum at around 33 degrees. But interestingly, we clearly thought MOI has a clear impact on the final titer. But in the end, when we had the results, the impact of the MOI was, let's say, minimal. Because no matter if it's a 0.001 or 0.005, we got the same virus titers. And for the pH, we also had a good optimum at pH 7.3, but the impact was also lower than we thought. But at that time, we have been quite happy. We scaled the process from the 1 liter up to the 50 liter scale. And during the COVID pandemic, also together with our colleagues in the US, we further scaled it to the 2000 liter scale with exactly the same progression of virus kinetics. So we were really happy on that. But then we went more into the data and thought, well, it took us for that specific project way more than 40 bioreactor runs to get a somehow solid process understanding. And then we thought, and that's where Novasign kicked in, we wanted to re-evaluate the whole experimental setup and see, couldn't it be possible based on all the knowledge we generated, and if we make use of the knowledge we generated, if we could get the same quality of data with a heavily reduced amount of experimental runs. And Max will present you then the results on that. So our general future perspectives are using that advanced development approach really helps you because you have predefined process blocks. You have quite good knowledge about the risk assessment. You have a quite good knowledge about your CPPs. And by having a good design and a good rationale in the design, how to set up your process, you can reduce your experimental runs. You can get a good databases with a reduced number of experiments, your process is getting more streamlined, your process is getting, you can get efficient, you have good knowledge. And you can do that not only for specific viral vectors, you can do that for every type of biotherapeutics. Yeah, that's it from my side. I think it will get more clear when Max is showing you the real data on the reduction. But yeah, I'm happy to take any questions if there are questions so far already. If not, I mean, we could also have a look at Max's presentation and then combine the questions.
I think we should have a Q&A now, and let's
Yeah,
see
sure.
so let's we can give everyone the opportunity to write the questions in the Q&A or raise their hand if they want to. But I have a quick, very specific question on slide 18, where you showed the microcarrier density impact on the virus titer. I mean, do you have an explanation for this local optimum that you observed here? Do you know what the driving force is?
Yeah, it is more or less that we then figured out. I mean, the whole information, it's not fully clear, but it seems that this specific virus type is forming an equilibrium between virus that is in the supernatant, that is budded from away from the cells, and virus that is still cell associated. And if you have very high cell densities, the gradient between virus that is still cell associated and the supernatant is less favorable of further budding and releasing virus particles to the culture media. We now just recently in a client project with a similar virus found out that if you treat the cell pellets with trypsin, for example, you can release virus that is still cell associated to the supernatant again, and thereby you could theoretically also increase the microcarrier density and cell density a bit more. But it's a very thin balance you need to take, especially what I mentioned before: more cells, more process-related impurities, and
the use of trypsin. Does the use of trypsin generate a product-related impurity?
No, not the use of trypsin. But for example, if you think you can now then also increase the cell density and the microcarrier density to generate even more virus, this then heavily complicates the downstream again, because you have almost double or triple the amount of host cell DNA and host cell impurities in your system. And your DSP train needs to cope with that.
Okay, and have you tried? Can you produce this virus type in suspension culture as well?
No,
you cannot.
I mean, there is a suspension virus cell line. Available. I mean, it's not commercially available.
Have you compared it?
We wanted to compare it, but it was quite difficult to get hands on that cell line. I know from our former colleagues that they tested the cell line. The virus titer was somehow comparable, but the problem is that the growth of the cell line is not the best, and there are speculations that this is not really a virus cell line anymore because it's contaminated with something else. So regulatory-wise, it's still the adherent virus cell line for that specific.
Okay, that makes sense. So if you would use the new suspension cell line, the regulatory pathway would not be clear, I guess.
Yeah. So
that's a big drawback.
Yeah, I mean, that's in general a point, what we are always doing when we are starting projects with our clients. We are often asked from clients, can we use that specific modern technique? Can we use that excipient? Can we use that type of media they recently saw at the conference or whatever? The problem is that many of the products really sound interesting and have a huge potential, but they are not available at the quality that is required for GMP. And if you are, most of our clients, most of the projects we are doing at the moment are for going into the clinics for clinical entry. And if I have some materials I cannot source in sufficient amounts, in good lead times, in GMP grade quality, sorry, I cannot use those materials immediately. So
Yeah.
there is always the gap between what's theoretically possible or what's available and what can really be implemented in a manufacturing campaign.
Yeah, there's always a gap between innovation and what is
Yeah.
actually realistic for biopharmaceuticals,
Yeah.
unfortunately. Yeah.
Yeah.
Okay. Well, I hope there will be more questions for Didi, but I think it would be a good time to hand over to Max to see the rest of the story. Yeah.
Mhm.
So with that, I will introduce our next speaker, Max Krippel. He's the head of consulting at Novasign. I'm very happy he can be here today, since he recently became a father and is now on a special on a special leave to quickly present and show us the Novasign side of the story. So yeah, with that, I hand over to Max. Thank you for coming.
Yeah, thanks for having me. Thanks for the intro. Yeah, I hope you can see my screen and you can hear me well. And I will show you, I will continue where Didi left off. So maybe a couple words first to Novasign, who we are, what we're doing, and then I will jump into the showcase we did with Bespark. Generally, Novasign is a software company with the vision to exactly what Didi said, to streamline the development, to take the right decisions at the right time, to bring this somehow into a software, to help people to design the proper experiments and to make use of this data and the knowledge. So our starting point is always the data, where we're trying to generate those information as smart as possible, meaning doing the right experiments at the right time, also with the right analytics and right measurements, so we can save time and costs down the line. Our vehicle, how we transport those solutions to our customers and also use it, of course, internally ourselves, is our software Novasign Studio, where we can make use of this data. And what does making use of this data means? It just means that we can do things that you can do with regular DOE analysis tools, like, for example, optimizing and characterizing your process, but also helping in de-risking, for example, scale up. We'll know that if you scale up a process at 100-fold, you will get some phenomena you don't have, and considering those early on can be very crucial in de-risking and getting it right earlier. But also, really, this is a vision slide, so really to go to able to at one point have automated process control. So if a model knows how a process reacts on different changes, it should also be possible to control those automatically. Of course, we are not there yet. I'm not aware of any process that is controlled via a model or via an AI, let's say. But generally, what we are developing now should be able to be used at that stage also. Couple words on how we operate. So within our software, we have pre-built blocks. The blocks that we used for the showcase with Bespark are the blocks for bioreactors and bioreactor simulation and for smart experimental planning. So how can we, from the 40 experiments that Didi mentioned, go down and reduce those and maybe retrospectively reduce timelines? But also one important thing is for us to be really transparent, so you can use parts of this software as you want. As said, we used now the simulation tool for reactors and the design of the experiments, and really be able to get those informations out. If you, for example, have your software of choice with which you develop or design your experiments, of course, you can take that and then maybe just use the modeling application within our tool. A couple of words to Novasign. So Novasign is actually a spin-off from Bochum. It was founded in 2019. We had our first version of our software in 2020 and used that for quite a long time, entering biopharma, industrial biotech, cell and gene therapy, and also cultured food. And then decided, well, technology moves on so fast, so software technology in this case, with cloud and then computations and all of that, we have to basically develop it from scratch. And we're doing this since 2023. And the last milestone for us was that a couple months ago, we received a strategic investment from Repligen, which they saw potential in our tool to also advance model-based bioprocessing using their specific devices. Okay, reducing development timelines. So the whole idea here is to take a shortcut, and instead of maybe performing one big design of experiments where you maybe perform 20, 30 experiments at a time, and only then look at the data to see, where is my process actually run, where's the best conditions in terms of viral titer, in this case, for example, or reduced impurities. We want to use an iterative approach where we can glimpse into the knowledge that we have at each iteration. Of course, you're not doing those 20, 30 experiments all at once. You're doing this in batches of 4, if you have a TASCIP system, or batches of 12, if you have an Ember system, for example. But looking inside and maybe having a model of each of these stages, which gives you insights on where to go. Am I on the right track? Are some parameters that I think are important not important at all, and I can leave them out? That's really valuable information. So basically, to get from a very reduced set of experiment, as shown here, like something like a fraction factorial design, for example, so just 4 points, to something where iteratively you add points, for example, these blue dots here, to then get to an optimum, like symbolized here in this graph with in green. And overall, we saw that we can save up to 70% of experiments. This is, of course, a value based on experience. There's no mathematical formula for this. But this is what we saw in the past that we can reduce compared to, let's say, state-of-the-art approaches or in-house approaches at different customers. Very important here, that's something that is for us really important to mention. There is a difference whether from where you take process data when you talk about model training. Here I'm showing just reactors and chromatography columns, meaning that this is valid for all process units. You want to use really data from the left on the top. So you want to go from small scale development data where the quality of the data is high while the quantity is low. You want to reduce those experiments, right? But you can actually induce changes there as you want. You can reduce the temperature, you can increase the pH, right? If you would use only data from larger scale, like manufacturing or pilot scales for this, you don't have this variation. So you yourself and also the model you train, the data that trains on the data will not know how to behave outside. So actually the use of a model is very reduced. You really have to be lucky that one of those runs failed because of a wrong set point. But this is actually quite unlikely. So we always do those experiments in development, as we also did with Spark. Okay, and this is actually the core of the presentation. So the workflow that we are using in order to make this model-based process optimization a reality. So as I said, we're starting from a reduced experiment. So what we did with Spark is we looked at all the data they generated. In total, we identified 22 runs to be useful in this sense. We then brought them all together. So we developed our pre-processing pipeline to bring online data, metadata, offline data of all of those runs into one file and train the model with that. I hope you can see my mouse, otherwise.
We can see the mouse, but now we can see the pointer, right?
And we train a model on that. What we then do is within this space here, so within this cube, we then simulate all the conditions. So what would happen if I change one of those process parameters? And I would get that within the digital twin application that we developed. And now what I can do is to see, okay, the optimum that I got, is this something I already ran? Is this a process I already ran, or is this just a simulation? In most cases, you will not have run it because you have a limited number of experiments, right? So you would go back and basically perform those experiments, take this data set and add it to the existing data set and retrain the model. And with this, you can imagine like you're drawing a line here in this cube and optimizing for a certain response, the titer, for example. Or in our case, what we did with this Spark was the ratio of titer to impurity. So to also think of downstream and reducing the burden on downstream here. And then once you're done, or once your experiments, the number of experiments you can do, or your resources are finished, you would exit this loop and say, okay, now with this, I have, this is my new optimum that I got. And in all the cases that we worked so far, you got at least the same or a better optimum than you would with a regular DOE approach, because you have the option to move. And this is just the schematics. It can also happen that you're actually moving outside of this cube, so outside of your initial boundaries, which then also adds process knowledge. Good. So jumping into the data. So with this Spark, we looked at the viral vector process, and we started out with a platform process. So with the defined pH, temperature, and multiplicity of infection, with roughly, when looking at the responses, we look at titer, HCP, and DNA. These were roughly the values here. So titer of 6.8 was something we started with, and the HCPs was roughly below 200 microgram per milliliter, and the DNA a little bit above 300 nanograms. And the objective was to, on the one hand, generally characterize the influence of the performance, and secondly, to optimize for lower process impurities compared to titer. Now, if you look here, so these here are our main outcomes of the process without any model yet. So what we did here was we took 6 runs. So this was 6 replicates. So in this case, we had quite some data to compare what actually our standard process is, and we overlaid them and measured the standard deviation. And that's actually just from any modeling perspective, or whenever you look at data, really important to assess, are my replicates actually the same, or do I have already a lot of variation in there? Because if the variation of my replicates and the variation of the runs where I actively change temperature or pH are the same, it just means your process is not controlled. So whatever the model will give out, or whatever assumptions you take from that, statistically probably won't hold true. Okay, then looking at the experiments, as said, we started out with a total of 22 runs. I'm sorry for that. That should be 22 with 18 conditions. As said, 6 are replicates. And you can see this is the design: multiplicity of infection here, temperature here, and pH here. This came from the software JUMP. It was an optimized design, so meaning it's not one of those regular full factorial designs. So this is already with some process knowledge in there, where you say, I think temperature is more important than pH, for example. I myself am not an expert on those designs, but we took those designs and tried to reduce it. So what we did in-house, it was mainly my colleague Benedict, was looking on what of these points can we actually remove and still get the same error, still get the same information on the titer, on the host cell DNA, and on the host cell proteins and the DNA released. And what we came up with was a design where we said, okay, if we train our model on these 8 runs here, so basically maximizing the space between the experiments, we can get already a lot of information from the run. And so we took those 8 runs with 7 different conditions and tested it on 14 runs. So basically the difference between 8 and 22, and looked at the error of that. If this is possible, if this would yield similar errors, it would already mean a reduction of 64% drug development. And what we got out of here, so this is trained on all the 22 runs, is a simulation where we see a lot of lines here. So these are the cells per microcarrier, the virus titer in blue, or in light blue, and then we have the impurities, so the double-stranded DNA, the host cell proteins, and the LDH as an indicator for cell lysis, right? And we can see already that we generated quite some process understanding. So what do we see? As in agreement to what Didi said already, if we change the temperature, so if we increase the temperature, we actually reduce we reduce the titer. But the reason for this is, if we look at the time resolved, that somehow you speed up the process, like the cells grow faster, but they die also faster. And then what you see, you have this sharp increase in all the impurities because your cell dies, and basically all of these molecules, they come out of the cell and you have to get rid of them later on. So it gives you already some idea on maybe temperature is an important lever, and while the titer maybe doesn't change that much, we see a huge change in the impurities. But that's just qualitatively, right? So we compared it also quantitatively. So as you can see here, a little bit reduced. We have the cells, the virus, and the DNA in this case. And we're now changing pH, temperature, and multiplicity of infection, our simulation. We have on the left the model trained on all 22 runs, and on the right, the model trained on 8 runs. And if you see the sliders, they move at the same time, just to have a better comparison. And you see the models actually look, or the simulations look actually identical. So we can say from this that the model learns from those 8 runs, basically the same as it learns from 22. And what does this mean? Well, as said, we have an experimental reduction. I talked yesterday with Didier again and said, can you somehow say how much this is in cost savings? And he said, well, 100K for sure. Maybe put a larger sign because with work hours and so on, this is definitely more. But also in total, 12 weeks or 3 months of development time. So you would be 3 months faster and 3 months just to get the same process understanding, right? And what we can now do is we freed up some resources. We could do another dashkit run and say, now we basically have this, we scouted through that space. What if we want to optimize even further? So can we design a couple of experiments which should be in the range of the best conditions? And basically, one thing I didn't show you here, but this is just the visualization tool. So what you can do also, you can show these results in a tabular form in our lookup table, and you can simply say, rank all of those simulations based on the best ratio of titer to, for example, host cell proteins. So then you would get biggest ratio, meaning the lowest impurities for the highest titer. We did this for the entire design space. We then used a space filling design in this section, where the model identified that the titer is highest while the impurities are lowest. So this means roughly at the center of the pH, at lower temperatures. So we see temperatures has a huge influence. That aligns with what we saw before in the previous presentation, and with a somewhat bit broader range of multiplicity of infection here. So we ran 3 experiments. One was, of course, a replicate. And basically what the results are, so this is how the and the DNA and the virus titer look like on our standard process. And we can see when we look, the process takes 160 hours. So what we can see now, if we add here in the dashed line in blue, so this is our optimized process. What we see is we get a similar titer. So these are the processes run in this new rectangle that I showed you. But we get way lower DNA, somewhat lower HCP, and also, since less cell lies, also lower LDH. Not only that, but we can also show that it doesn't really, we don't actually have to run until 160 hours because the impurities, they increase exponentially almost at the very end. So if we stop this process 24 hours earlier, we would maybe get a small reduction in titer, but a huge reduction in impurities. And when we think of that in production, this process will be run all the time, so in shifts. This means you actually save a day there. And with this, you also, if you look at the space-time yield or the volumetric productivity, it would further increase your output. Okay, with this, I'm at the end of my presentation. So looking at this, a model-based DOE is an alternative to the state-of-the-art DOE as done in industry almost, I would say, everywhere, and allows to design new experiments in the same development campaign. In this case, the process model learns from 8 runs instead of 22 runs, and you could really learn the same or get the same process understanding and free up some resources to do other experiments, basically optimizing, maybe even go out of the space. In this case, the space was set correctly so that we say the optimum lies within here, or at least our local optimum. And generally, this is a more streamlined way to optimize processes in general, but especially for cultivation processes. Maybe one thing that I want to mention here is, of course, this loop of evaluating a model on the current data, simulating it, and deciding for the next experiments. If you start this the first time, or if you do this the first time, you will take more than just a couple of hours to set this up. So it is having a streamlined data import and pre-processing of all the data. We're talking offline data from different devices, I think at least 4 devices here, online data, analytics, that's really important to make sure that this loop that I showed you before can really be done rapidly. And we try to provide tools for that, for our internal, but also for our customers in order to make this a reality. Okay, with this, I'm at the end of my presentation. Thank you very much for your attention, and I'm happy to take questions.
Great. Thank you, Max. Really nice presentation. I think I forgot to thank Didi earlier. Yeah, so also, Didi, thank you for this nice presentation. Very nice. I hope we already have a question in the audience. Okay. This is a question for Didi, but let's start there. Yeah. So Leo Gunderson asks, in the risk assessment part with the various impressive boards, did you gather the possible risks and correspondent probabilities from your own experiments or from available data? How long did this process take? Let's start there. There's also another question for Max.
So you were actually asking how long the risk assessment itself took, right? Or did I get that right?
Leo, if you want, you can also. Yeah. Okay. He says yes. Yeah. So how long the risk assessment take? If you want, you can also raise your hand, and I can call you into the onto the floor. If you want. But okay, Didi.
Maybe I will go. So in the risk assessment, what we did, we took only our own data. So all the risk assessment was based on the own data, on the own process data that were already available, and this took us already quite a lot of runs. I'm not even sure how many those were, but we really had all the process knowledge we generated so far, and then sat together in the room for 3 days as this one specific risk assessment. We locked ourselves in a windowless room in Hilton Paddington in London, together with some consultants, and then really went through every single step of the process. So ideally, you have a base process already developed, because you can of course also do a risk analysis totally from scratch, from the greenfield. Think of ideas on your process and what could impact the development, but we based everything on our own data. Yeah.
Okay, I hope that answers the question from Leo to Didi, but there's a second question from Max. How do you choose the 8 runs that build the model capable of representing the 22 runs? So this is also a question I wrote down. Yeah, so maybe that was on slide 12, Max. I don't know, because maybe I missed it, but how did you pick those 8?
Yes. So at this stage, this was a rather manual process. Generally, the framework for this is to space. We know that our model. So generally, those machine learning, our model, we're using hybrid models. So which trains on the one hand on process data, but on the other hand, and that's important, also it always includes the previous iteration of what was predicted. So actually, we know where we are in the process. So we're doing a time resolved. these models are really good at interpolation. So if you have 2 points and you simulate in between, there's a very high chance that you get the trend right. And what you should really avoid is trying to go out of that region. So it might go well if you go maybe half a centigrade out, but don't think that if you simulate something at 50 degrees that it would actually represent anything that anything useful. So the idea was to space it as much as possible. At that point, we did this manually, and in parallel, we wrote a tool where we set where we basically import those 22 runs and say, I want X runs, in this case 8, and I want the distance between those points. So there are things like just to make the easiest one would be the Euclidean distance. So really you have a 3-dimensional space and you look how far they are apart and you maximize those. So it's an optimization basically. And of course, if you say I want 8 runs versus I want 9 runs, this will be completely different runs, because by adding 1 point in between, all of a sudden the distances, the overall distances, they change. So that's something, a field for us to investigate even further. We have some basic tool for internal use. So they're not in our software yet because they need a lot of human oversight still. But that's the general idea on how we do that. So we know our models can interpolate, therefore we're trying to remain within that space and trying to space them, these points, as far apart as possible, which is also the same approach when we designed those 3 experiments to do the optimization.
Okay, Leo, I hope that answers your question.
Yes, thank you very much.
Okay,
Welcome.
then I have another question. So actually for both of you. So if you start now with a new project and an unknown product, yeah. So how do you choose the starting point for the novel process development, and how can you be faster using this methodology?
I was actually waiting for that question. In general, I mean, what we presented here was based on a baseline process that was already established, and how you can, if you have a certain framework, if you have a platform established and you want to try to bring a new product out from that platform, how you can save it. But in general, if you're starting from the green field, and that also happens quite often, keeping all the principles I mentioned before of really setting up a QBD-based strategy. Think about the main process parameters. Think about the CQAs as a basis, and then maybe also based on literature, based on prior experience, you could think of, because I mean, many processes for antibodies or for viral vectors itself, they're running in a, let's say, similar kind of framework. So, and when doing the development, even in very small scale, in shake flasks or whatever, you already get a little bit of feeling about what could be an impact. And then you do a very basic risk assessment, very basic, and think of 2, 3, 4, 5 potential CPPs without a lot of all detailed knowledge you generated yourself. And based on that risk assessment, you can then take those parameters, feed them into a possible design, and then do, as Mark said, an iterative process. Because with a classical way, we needed 40 experiments to get to a certain point. If you're doing, let's say, the model-based DOE design, you have, based on the risk assessment, you do your first run, see the outcome, feed the outcome into an iterative process, do the new design, suggesting you which points could be done next, and then you're iterating yourself to the optimal point, with maybe not such a great reduction of the 67%, but in the end, generating even more process knowledge with a, again, still reduced number of experiments. And that's something we are still at the moment fighting with discussing with clients, with people that start the process development, because they mostly say, well, I just need something that works for clinical start. And we had that going into a project, just something that works for clinical start, and we're getting data and we can be bought by a VC or whatever. But this is not a solid foundation, because if you have just something that is just good enough for that, you cannot build on that. And if you invest a little bit in the beginning in a thorough design and lay a good foundation, you can use that basis for building up and generating more knowledge and more knowledge and more knowledge in an iterative process. It's not necessary to do everything from the beginning, but at least a solid foundation.
Max, do you have anything to add?
Maybe just a couple words to the data. So we are really we have a lot of experience in optimizing and characterizing there where some data is already there. If you're really starting from scratch and you'd have no previous knowledge, probably we will not be able to help a lot there, or modeling in general. What you need there is to say the same way an expert thinks about it. In my previous let's say I have a map and I have already developed 10 maps, and there we always used this and that conditions, and that worked okay. And then we started from there. You have to put this into numbers somehow. If you're able to put this into numbers, so what makes them similar? Of course, they're all maps, but also what makes them different? Are there maybe some charge variants, or are the charges distributed separately? Do they have different PIs? I'm not a map expert, but something that defines how those molecules are expressed differently under the same conditions, and start from there. And that's an active field where we investigate, and we're trying to also go more into detail, because this knowledge is there, but it comes from different departments. And there is again this old saying of those silos. So when we are talking mainly to development departments or MSAT, but if we go into this if you want this information, we have to go to really to the cell line development. And companies are very keen on their cell line development data. They will not share this, not even anonymized, and that's something that I think we still have to work on. Yeah.
Okay, there's a new question from an anonymous question. So curious about how this plays out in cell therapy processes, which can be quite diverse, have lower number of runs, and include more manipulations in comparison to traditional vaccine or small molecules manufacturing. So I don't know if you two have any experience with cell therapy.
Well,
yeah? In a very exciting field. Yeah?
Well, I mean, cell therapy it's also a quite diverse and controversial field. I mean, cell therapy is also part of gene therapy somehow. It's just the ex vivo gene therapy. True. Mostly. Regarding the processes, I mean, when we're talking about whatever CAR-T, I mean, there is such a broad range of cell therapy. Many of the cell therapy applications are currently done by, the ex vivo is done by a viral reprogramming of the cell. Mm-hmm. And the viral reprogramming is done, for example, by lentivirus. And there you also have a process for producing a virus. So the process for producing the virus for cell therapy, there we are quite comparable to the settings and to the frameworks we have with vaccines or with AAV or whatever. The process itself of transducing the cells and then reapplying them to the patient, that's fully different. And this is also not our main expertise, because we are focusing mainly on the production of the viral vectors for transducing the cells for gene therapy. So I would say one could for sure think of implementing all those strategies and all those ideas, not only to the production of the cell therapy viruses, but also on how to transduce the cells, in which conditions, whatever, when to best reinfuse them into the patients. But I think I'm not an expert on that, and there is quite for sure a lot of work to be done in order to get that streamlined. But I think the basic principles are always the same: thinking, making thorough plans, and then starting the development, and not starting the development and then thinking and making thorough plans.
Yeah, so Max, I don't know if Nova-san has any experience already with cell therapy processes. But
Yeah. Well, not in the case of, so we do cell therapy, but not in the case of taking information from other cells or other products in this regard. We see this interest from that and customers asking for this for recombinant proteins and for MAPs. So that's something that we actively work on. But also I have to say, we're in a field where in a phase, I think, where we're trying to get those gene and cell therapy process stable, like reproducible, that all this graph that I showed, that we actually overlaid those 6 replicates from BSpark. That's actually what I forgot to mention. It's actually really, really good, because if we look at similar processes, maybe run in shake flasks, or where people say these are replicates and this is our basis, those vary more than any induced changes sometimes. And then there's not much we can do except for saying, invest in your analytics or in the training that these processes get more reproducible, because only then you can make informed decision. And that has nothing to do with modeling. It's just looking at the plain data and seeing already changes in your replicates there. So I think, to answer your question, I think we're still far away from making these decisions on that level, data-wise at least. And yeah, we'll have to get the processes reproducible first.
Maybe I can add something here as well. We're also talking about reproducibility and data and quality of data. I mean, when talking about live viral vectors, for example, for vaccines, all the methods for determining the viral titers are biological assays. And all those biological assays have an inherent variability of depending on how good your operators are, how good your procedures are, and how many replicates you're doing, of 0.3 logs, 0.4 logs. So the quality of the data itself already has a certain variability in there. And if the runs themselves have also the process runs have a big variability, and then the analytical methods add on top a big variability, then the question is how precise modeling can be.
Yeah, I mean, it would require a large number of replicates, right?
Yeah.
And I mean, cost prohibitive at some
just
point.
To get you an idea for releasing that virus I had in the case study, for getting a reliable titer, we had 12 replicates of 3 operators on different days in order in order to have a really reliable virus titer that is good enough for releasing. But also when adding something on data quality itself, we worked a lot with different CDMOs in the past, and we're now talking about model-based controls, model-based development, etc. We also realized that this is far away from being introduced into CDMOs, because, for example, when we back then asked, could you provide us the online data of a certain run? In one case, it happens that we got a picture of the bioreactor screen. This was the provision of the online data, because they were not able to store the online data of pH or whatever. So this is such a discrepancy between what is theoretically, technically possible, what could be implemented, but what we see in real world. This is, yeah.
Okay, thank you. There's a new question, mostly to Max. If one is just starting to do fermentations, at which point will it pay off to try adding a hybrid approach to lab experiments? How many and how diverse of an experimental data set is required to go into hybrid models to start with? So which process knowledge is already required to start? And then there's a second part also, how broadly applicable are such hybrid models in general? So transfer insights from one protein to another.
Yeah, so to answer the first question, well, you need somehow a good starting point. So basically, something where your process where your process actually performs, produces your product and performs, and you can move on from that point. Then I'd say the first optimization or the first screening you do there, like that you can find in literature a lot of those, whether it's a recombinant product, whether it's a microbial based an antibody, cell and gene therapy, what factors drive your process. This is at least the common ones. This is easy to find. And I think from that point on, so hybrid model will not give you any insight in whether you should first vary temperature or dissolved oxygen. But from that point on, you can actually replace the DOE, so basically the models in DOE, so MLR or OLS, for example, with hybrid models, because you will get the data you train on is basically, or the runs you train on are the same. One big difference is you have to do time resolved. Everything you measure and everything you want to simulate should be time resolved. So it's not sufficient to just measure your product or your titer at the very end. You have to do this several times. So on average, what we tell our customers is 3 to 4 times per run. That gives a good basis. And there comes, of course, miniaturization as a problem. So maybe if you run it in number 15, maybe you cannot do that. So you will have to go a bit larger, or what you do is you have to parallelize and sacrifice different runs. But that's so going very small can be one issue there. But from that point on, actually, you can use it as a guideline. You can start out with, I found on literature 7 potential CPPs that I change. You do the first screening and you realize in the model that basically if you change 3 of those, nothing changes. So it might not be critical for you, or you say it's very robust. That's the way to say it. It's very robust on the changes you do, and you will not influence that. And by this you can then reduce quite a lot the experiments down the line. And regarding the knowledge, it's what is needed. It's similar to what I said before. If you can pinpoint it to some characteristics of the process or previous products, and say, I know my products have different quality profiles, and I take this into account. So I take my previous 3, 4, or 10 products that I developed into the training set, and then I can use this and I know that the attribute of my 11th product, I can include that. The important thing that's my personal opinion, if it can somehow be done numerically, so not categorical, but numerically, so saying maybe the PI, so really a value that is now I forget the word. It's not discrete, it's continuous. That makes it easier. And of course, the more the modalities are well established, the more of these analytics you have, the more of these analytics are physics based and not biology based. So basically, they're also more reproducible, and you can do them easier and more automated. So that all of these things of PAT and analytics and real time and so on, this all plays a role there.
Maybe I can add something here as well, because that's a question we're also asking, or also asked by clients. And if you're starting from scratch and you're applying all those concepts, you might not be way faster as compared to another classical one factor at a time approach, or when you're just starting, but you're definitely generating way more process understanding, and you know your process way better. So if something fails during a production campaign, you might know why it failed, and you take counteractive measures. When you don't have that process understanding at the beginning, something happens, you can just, try to do some root cause analysis and go back to the start and just redo it. But gaining process knowledge is, I think, a very, very big asset.
That's fine. Thank you. So before we go very much more over time, I would also like to thank Victoria Zangelini for being in the background and making everything work well. And with that, I think we can stop the recording and then continue with other questions.
Mm-hmm.